Hello, welcome to Cyrus Bio. Bench has a wide variety of features, but we want to get you started with the simple one, the tool to improve the binding interface between two proteins. This is one of our most popular tools, and it improves interfaces far better than any other software tool. So what you see here is the Bench platform when you open it. I'm going to show you how you import a structure shown here. We're going to import an antibody-antigen complex. We're going to look at it, we're going to accept it by clicking the button here, and you'll see that it comes back. That spinner turns into a check mark. Once that happens, we can do further processes on that protein complex. So I'm going to open it up. I'm going to show it in the workspace at the bottom. I am going to show you how you would select the H3 loop of the antibody here. And I'm going to look at the sequence viewer and select the residues of the H3 loop right here. And once I've selected those, I'm going to create a selector by clicking this button over on the left. That means we have the H3 loop saved, so we can use that subsequently in our design process. Next, I'm just going to color the protein complex, the antibody-antigen complex, so that it's a little easier to see the antigen at the top and the antibodies at the bottom. So after this, the next thing we need to do is prepare the structure. So to do that, we're going to go up to the upper right-hand corner of the panel and click the prepare button, which I'll show you here in a second. All right, so we click the prepare button, and the program's going to go off and do a restrained minimization of the protein, basically, to get it consistent with the Rosetta force field. And you do this whenever you bring in a PDB file. So there's a prepared protein. The next thing we need to do is do a design experiment now on the H3 loop. You see I have that selector for the H3 loop, which is also shown in red there. So I'm going to go over to the right and pick Flex Design button right there. And once I pick the Flex Design button, I'm going to pick the selector, the H3 loop. I'm going to tell it I want to do 100 iterations of this, 100 iterations of about 1,000,000 moves each. And then I'm going to go down in this panel and tell it what kinds of mutations I'm going to accept. And in this case, I'm going to accept all the mutations except for proline and cysteine and glycine, because those typically are not mutations you want to introduce. And once I'm happy with that, I click this Run button shown there. The program's going to go off and run that, and in maybe about 30 minutes to an hour, it's going to come back. What we will get back is information on what types of mutations are acceptable and at what positions. These can be viewed as a logo plot as shown. These are mutations in the H3 loop that are predicted to improve the binding of the antibody to the antigen. The predictions can be used individually or in aggregate to focus a screening library. Now we encourage you to go back and learn more about Bench at support.cyrusbio.com in the Getting Started section, or just jump in and get started.